In this video, we will cover the last subtopic in chapter 4, that is genetic mapping. We will see what is meant by genetic mapping, and then we will do some calculation. By definition, genetic mapping, it is a technique that we use to study the position and also sequence of genes on one single chromosome. So this means this technique, it can be only used or carried out if the genes are linked and also crossing over takes place. If you remember linked genes, so this refers to genes that are located on the same chromosome. And depending on their distance, okay, so two or more genes, they have higher chances to be inherited together. So that will produce parental offspring, in which offspring have the same characteristic or trait as the parent. And second, crossing over here, this one is referring to the exchange of a portion of the chromosome between non-sister chromatid of homologous chromosome, okay, during meiosis, to be specific, prophase 1, and the product here is to produce recombinant offspring. And recombinant offspring here, so this means the offspring, okay, for a gene or for a trait, it has recombinant of allele from both parents. So for the linked gene on the same chromosome, okay, so let's say the first one, the distance of the genes, they are close to each other. So this means this linked gene, they might not be separated during crossing over, which is occur in meiosis. So we will produce gamete with parental combination. Okay, so that's the first situation. So the second one, let's say these genes, although they are located on the same chromosome, but the distance is quite far from each other. So during crossing over, these genes might be separated, okay, during crossing over. So you will produce offspring with recombinant type. So that's why if you can see the last sentence here, so the crossing over frequency is directly proportional to the distance between the two genes. So the higher the crossing over frequency, this means the longer the distance between two genes on that same chromosome. So there is the higher percentage of having recombinant type of offspring. So to find out the distance between two genes on the same chromosome, or what we call as here crossing over frequency, so we have this formula. So let's say we know the total number of recombinant offspring from that cross, and then total number altogether of offspring produced and times it with 100. Okay, so we can find the crossing over value. Remember that the crossing over value is also equal to the distance between two genes on the same chromosome. So that's why by convention, let's say 1% of crossing over value, it is equal to 1 map unit. So map unit ni ataupun centimorgan, written as C capital letter M, bukan C M kecil itu sentimeter. Untuk centimorgan kena guna C M besar. Okay, so we can write the distance between two genes using this unit, okay, MU ataupun CM. Antara dua ni boleh guna. Okay, so sini ada penjelasan sikit. So let's say the genes are not linked, okay, as in they are not on the same chromosome. So that means the phenotypic ratio of the offspring will be 1 to 1 to 1 to 1, as in the Mendelian inheritance. And also, there will be also 50% chances that the alleles on separate chromosome will be inherited together. Okay, 50% and above. So that means, okay, so kalau COV is lower than 50%, so maksudnya the genes are closer. Okay, and paling penting, they might be on the same chromosome. So this here, okay, this diagram here shows how genes are mapped on a chromosome based on recombination data. Okay, so here, let's say gene A is here and there is B. Okay, so as you can see, the distance is quite close to each other. So that means the crossing over value K is also smaller. And here, if you compare with H, so H is the most further. Okay, so the crossing over would be bigger. we are done with the theory part. So now let's apply the knowledge to find the crossing over value. So here is the example 1. So as usual, soalan ni, mana-mana soalan dalam chapter 4, dia memang akan bagi info panjang-panjang begini. So make sure you read bersabar, baca je dulu satu persatu, jangan rushing mau jawab soalan, no need. Take it easy and do it calmly, Dan yang paling penting untuk cari crossing over value, memang soalan akan bagi jumlah, amount of the offspring. So sini kamu kena cari mana yang recombinant, mana yang parental, So, kalaupun kamu tak faham explanation saya yang panjang-panjang tadi tu dengan yang sebelum ni, untuk parental, kamu ingat parental dia punya nombor tu besar, So, biasanya ada 4 nombor. Kamu cari je 2 nombor yang besar. So, itu adalah parental phenotype. Dan 2 nombor kecil, that is recombinant. Itu tips pertama. Soalan ni dia tanya, what can we predict from the data relating to the distance of the genes? So, kita tengok dia punya ni, explanation. So, macam yang saya cakap tadi, so based on the numbers of the offspring given, kalau 2 nombor besar, that is referring to the parental offspring. So, parental offspring ni maksudnya offspring yang ada trait sama macam parent. Either one parent. Lepas tu, nombor yang kecil, these two, so that is the recombinant offspring, jumlah recombinant offspring. Offspring yang dia punya gene tu mix, mixture of 2 parents. Itu satu. Lepas tu, from that data also, we can conclude that the genes are linked, because we have high number of parental offspring. Macam mana? So, ingat balik, kalau genes tu bukan link, they're not on the same chromosome, the ratio of the offspring, it will be 1 to 1 to 1 to 1, as in the Mendelian inheritance. Tapi di sini, mana ada ratio 1 to 1 to 1 tu? As you can see, ada 2 nombor besar, 2 nombor kecil. So, itu maksudnya the genes are linked on the same chromosome. Second, crossing over occur. So, walaupun genes tu link on the same chromosome, tapi crossing over occur. So, maksudnya at some point, the genes are separated. So, macam mana kita tahu genes tu separated? Macam mana kita tahu crossing over occur? Because, you have the recombinant offspring. Kalau recombinant offspring ni, we will not have recombinant offspring kalau genes tu link and no crossing over occur. Sebab kita cuma akan ada parental offspring. Tapi di sini, we have both parental offspring and also recombinant offspring. So, that tells us crossing over still occur, although the genes are linked. And here, we can find the crossing over value. Ini guna formula. So, total recombinant offspring, bahagi dengan total offspring. So, ni kamu tambah dengan ni. Lepas tu, bahagi dengan jumlah offspring semua darab 100%. And you found out that is 20%. So, maksudnya di sini, distance between the R and S genes on the same chromosome is 20 map unit ataupun centimorgan. So, ni boleh guna M.U ataupun cm besar. So, biasanya untuk soalan macam ni, jawapan dia hujung sini mesti ada statement. So, the distance between the two genes is divided. Itu untuk soalan begini. And here is example 2. We are given genetic diagram. And here, memang untuk soalan genetic mapping ni, dia memang dia memang akan bagi jumlah. And ingat, nombor besar, parental phenotype. And then, nombor kecil ni recombinant phenotype. Ni offspring dia punya genotype tu combination from both parent. Kalau parental ni satu sama dengan parent 1, satu lagi sama dengan parent yang kedua. So, based on this question, based on this information, find out the crossing over value. So information yang kita ada, parental combination, nombor besar tadi, lepas tu recombinant, nombor kecil. So, COV value. So, untuk soalan macam ni, tips dia, pertama sekali, you just write down the formula. Kadang-kadang, depending on the year, dia kira sekali, formula tu satu markah. So you just write it down. Total recombinant offspring. Kalau skema tu tak bagi jawapan untuk formula pun tak apa. Okay? Tak ada efek. Yang penting kita just tulis je lah. Ni untuk paper tu lah. Okay? Lepas tu sini total offspring. Times with 100%. So sini is the formula. So total recombinant nombor kecil. Lepas tu bagi dengan total offspring. So nombor kecil tambah dengan nombor besar. Okay. Tambah dulu. So 40. So semua ni 1000. Okay. Lepas tu kira terus. So your answer should be 4%. Lepas tu statement. Okay. So the distance between C and S is, jangan tulis 4% lah, tapi guna unit 4 mu ataupun centimorgan. So here we have example 3. So it says here that P, Q, R, and S, perlu dan di sini, genes are linked. So the crossing over value for pairs of gene, they are given here. So this question, it asks you to map the chromosome. So the tips or hint here, you start with the furthest. Maksudnya those that are pairs with highest COV. Maksudnya di sini, okay, that pair of gene, they are the furthest from each other, yang paling jauh. So maksudnya R, S adalah di tengah-tengah P and Q. Okay, so mari kita tengok dia punya jalan penyelesaian. So first you rearrange of the pairs of gene with the decreasing value of COV. So yang paling jauh tu kita letak paling atas. Okay, so we start with this first, R and Q. So R di sini, Q di hujung. So altogether 40 map unit. Lepas tu P and Q 35%. Okay, so maksudnya P tu dekat dengan R sebab distance between P and Q 35. Okay, lepas tu Q and S is 10%. So maksudnya dia closer, S ni closer to Q. Lepas tu P and R, okay, ni paling last, P and R is 5%. So maksudnya P is close to R. Okay, so altogether macam ni. And lastly, to conclude this subtopic, so through genetic mapping, we can determine the distance between two genes on the same chromosome. We also can find the linear sequence of the linked gene on a chromosome. So this would help the study of genetics. So this means we have reached the end of this chapter. So before we finish, okay, let's try a partial question. Okay, so soalan partial kita ada dua. So kita sambung dengan yang 14C di sini. So macam yang saya bagi tahu sebelum ni, first baca dulu information yang soalan tu bagi. Fahamkan dulu apa yang dia bagi tahu. Okay. So sini heterozygous fruit flies with a black body and purple eyes were mated with homozygous flies that have a grey body and red eyes. Okay, so they produce 1000 offspring. Okay, so ni dia bagilah jumlah semua ni. Lepas tu dia suruh calculate the percentage of recombinant types of offspring. So yang ni dia sudah terang-terang bagi tahu. Okay, dia mau percentage of recombinant type. Ni tak payah pusing-pusing fikir. Okay, dia sudah bagi tahu. So 120 of the two. Ya. So untuk recombinant types kita ada 120 all together. Okay. So terus cari macam ni. So you have 120. So di bawah ni total offspring 1000 tadi sudah bagi tahu. So you just write down straight away 1000. Lepas tu darab dengan 100. 100%. So you have, sorry lah, my maths is kind of rusty, so perlu guna calculator. Okay, so that is your answer. Okay, and then the second question, determine map units between the body and the eye color loci. So maksudnya dia suruh cari distance between the two genes. Okay, so since you have calculate the COV di sini, okay, so ni basically COV lah. Okay, kita guna formula yang sama juga. So by convention, so 12% here, so that is also 12 map unit ataupun centimorgan. Okay, next we have question number 10. So this is the second question that we discussed in this video. Okay, as usual, read the information first. So in maize, jagung, a gene for colored seed and smooth seed are dominant over the gene for colorless seed and also wrinkled seed. So pure breed strands. So kalau pure breed ni maksudnya homozygous dominant dengan recessive. Parent satu dia homozygous dominant, parent kedua dia homozygous recessive. So a test cross for F1 generation produce the following result. So maksudnya di sini, okay, parent dia, okay, yang ni pure breed. Lepas tu dia hasilkan F1, lepas tu F1 to cross, ni adalah kita punya F2 generation. Okay. As you can see, based on the numbers here, so kita tahu sudah ni adalah link gene. Okay, sebab dua nombor kecil, dua nombor besar. So nombor besar ni parental phenotype, nombor kecil offspring, recombinant phenotype. Okay, so cara tulis ni sebab dia link gene, remember. So first parent nenek lah, nenek yang ni, nenek datuk nenek yang ni. Okay, yang asal tu pure breed. So you should write it macam ni untuk dia punya genotype. Slanting line means link gene. Ni parent satu, lepas tu parent kedua, semua huruf kecil. Okay, so straight away you know F1 punya genotype macam ni. Okay, so the test cross, so kalau test cross maksudnya dia cross ataupun breed dengan individual that is homozygous recessive for both trait. Okay, so ini information. Okay, based on the analysis that you done on this question. So let's go to question A. So question A, does the above cross follow the Mendelian ratio? So ni referring to the test cross. Okay, test cross between F1 which produce F2. So kamu tengok sini ratio dia 1 to 1 to 1 ke? Okay, so your answer is no. Explain how the situation in A1 occurs. So kenapalah kamu pilih no? Dua markah, so you need to give two reason, okay, ataupun 2 point. Oh, ini no tadi untuk ni. Lepas tu 2. So how do you know they don't? Why they don't obey Mendel's law? First, okay, the genes are linked. Boleh tambah on a same chromosome. Okay, satu lagi due to the presence of parental and also recombinant offspring. So maksudnya how you are able to get a recombinant offspring? Okay, that is because crossing over occur. So that's why you don't get that 1 to 1 to 1 to 1 ratio. Okay, that is for question A, and then B. So he asks you to draw the genetic diagram for the test cross of F1. So that means you don't start with P. Dia mau test cross F1 sahaja. So that means you start with F1. And from our analysis, we know their genotype. Okay. Tapi to make it clearer, so kita guna page sebelah lah. Okay. In case you're not sure how do I get that answer. So ingat from our analysis, P dia tadi ni belum lagi jawapan lah, so just small show. So the first parent is homozygous dominant for both traits cross with second parent that is homozygous recessive. So the gamete from this parent, okay, ni since both the same, okay, on homologous chromosome, the same gene, so this parent will produce the same gamete, okay. So arrow lupa pula, arrow and then circle. So from this parent pun satu jenis gamete. Okay, and fusion occurs, so the genotype of the offspring will be heterozygous. Okay, so this is your F1. Okay, tapi soalan B tadi dia tak mau dari atas ni, dia cuma mau test cross for F1. So that's why for your answer, kita letak di bawah sikit lah supaya boleh senang nampak. So you start with F1. So you write down the genotype. Okay, test cross. So the partner or the second parent is homozygous recessive. So gamete, G gamete. Okay, since we have different, okay, this individual is heterozygous, so this individual will produce two type of, not two. Since we know crossing over occur from the data, okay, so we'll have 4 different combination of the alleles, okay. So kalau link gene without crossing over, dia cuma akan produce two type of gamete. Tapi di sini, since we know crossing over occur from the data, dua nombor kecil, dua nombor besar, so this individual it will produce 4 different gamete with different combination of alleles sebab crossing over occur. Okay, so ni first possible combination, and then silap ni F besar. Lepas tu ni combine dengan yang itu, F kecil. Ataupun C kecil combine dengan F besar. Dan C kecil combine dengan F kecil. Okay, so 4 possible combination for the gamete. Meanwhile, for the homozygous recessive parent, so sebab semua allele sama, so the gamete only have one type. Okay. So for this one, you can use Punnett square if you want. Kalau tak mau pun tak apa sebab dia tak specify. So your F2 generation, okay, so the the arrow here, that means fertilization occurs. So here you will have heterozygous offspring. Ni dengan ni, C F kecil. Okay. Dan ni dengan ni, C kecil, F besar, semua kecil. Dan ni homozygous recessive. So kalau kamu perhatikan, okay, ni parental phenotype, okay, dua ni parental phenotype, dua ni combination, okay, ni kombinan lah. So that shows the crossing over occur sebab ada recombinant phenotype. So soalan B ni, okay, the marks given is 4. So the correct genotype for F1 1 markah, then correct gamete 1 mark, and then the correct genotype for F2 is 1 mark. So untuk markah terakhir, you need to state the phenotype. Tapi jangan bagi ratio, okay? Ini bukan Mendelian. Kalau Mendelian boleh bagi ratio, tapi ni jangan sebab dia punya ratio bukan 1 to 1 to 1. Okay, sebab dia sudah bagi data tadi tu, so you don't have to include ratio. So you just write down the phenotype. Okay, so write down the phenotype. So this one it will be, sebab ada satu dominant allele, so this one will be colored. And the satu F dominant allele here, so it will be smooth. Sini pula also colored but homozygous for the F ni, so that is wrinkle seed. Okay, ni pula colorless homozygous recessive kan, tapi ada satu dominant allele, so you have smooth seed. Ni pula colorless, ni homozygous recessive for both thread and wrinkle. Okay, ingat tak payah ratio. And the last question, C. So kita balik guna slide ni. So C. Okay, calculate the distance between the two genes. Okay, so show your calculation. So ni kita guna formula COV. So equal to, biasakan diri tulis je full formula tu. Darab 100%. So ingat dua nombor kecil, 14 plus 10 bagi semua total. 80, 396, 14 tambah 10 times 100%. Sekejap saya kira dulu. So, the answer is 3%. Okay, untuk ni, ayat last dia. So, kalau boleh tulis ayat penuh. The distance. Three map unit. Ataupun, pilih satu cm besar.